Okay. That's me and secret. This is how do it work? Yeah, quite intuitively, yes. And also, you can see your presentation here on the screen. So it's a pleasure for me now and to moderate together with Siegfried de Kasmaker from the Belgian Science Sano, the next session, which continues about validation and standards. And I think as we are a little bit in delay, about 20 minutes, so I don't want to spend too much time, but I think we already discussed also in the panels some important questions about the in silico standards and so on. So it's a pleasure really now to introduce as a first speaker, Professor Marc Elroy, who retired recently from leading a lab at the Institut Pasteur in virus discovery. And I don't start to summarize all his academic benefits in work, but I just to mention, he also founded the company PathoQuest, and he was into the NGS field really since very early, since decade. And now really we are keen to hear the approach about the transcriptomics that we did not discuss that deep so far. Thank you very much.
So thank you for your invitation and presentation. And I'd like to share with you results of experiments supporting the replacement of classical tests, meaning PCR, HAP, MAP, RAP test, and 9 CFR assays by NGS transcriptomic, and which is validated at the GMP level. And in the second part of the talk, discuss how the results can inform the safety of testing of cell banks, routine testing of non-purified bulk of recombinant proteins, and testing of CAR T cells. So briefly, you know that when a virus replicates in cells, it accumulates a lot of messenger RNAs. So that's the target of transcriptomic analysis. That's different from viromics and genomics that target DNA or RNA genome. So why are you using this transcriptomic assay? In fact, in comparison with the diversity of the genome associated with virus particles, there is a common molecule, which is messenger RNA, which is common to DNA and RNA viruses in infected cells. It's single-stranded, it's a positive strand. It's expressed during the replication of all viruses, including the DNA viruses, indeed. The level of expression is often very high, so it's easy to detect. And it's also easy to detect because highly expressed RNA from the cells correspond to short sequences that could be eliminated, and that's not the case for DNA. So this increases the ratio between the target and the background, and this increases the sensitivity. And in addition, regarding now the interpretation of the results, analysis of RNA profiles or metabolic labeling, we have published that, of nascent RNA can help for the interpretation of the result, because the pattern of transcription could be very useful to distinguish real replication from carryover of proteins, of sequence. So the objective of the study was, we have previously shown that this NGS transcriptomic assay can replace in vivo test. We have compared 9 different viruses with corresponding cells and in a head-to-head comparison with in vivo. And now the context, as you know, is that ICHQ5A allows NGS, targeted or not, to replace virus-specific PCR assay and rodent antibody test. And the question is, can this NGS transcriptomic assay can replace this test? So question, what is the analytical sensitivity? What is the ability to detect distant strains? And then a discussion about performance for the testing of the 3 samples I have cited. So the virus selection, so obviously the tests we want to replace are targeted tests, targeted against a list of viruses. Regarding bovine and porcine, they are described in 9 CFR, and you have the list here. Regarding the rodents, they are described in ICH. You have the list here. And regarding humans, there is a typical list of viruses that must be tested. And this is a sum of 78 different viruses we have used for this evaluation. What we have done corresponds to first the test samples. So regarding rodent viruses, we have made two kinds of pool using synthetic RNAs. So we have used synthetic RNAs. One pool corresponds exactly to the sequence of the reference strain, plus a negative control, which is a mirror RNA, meaning an inverse sequence of one of the target, plus distant synthetic RNAs. I will explain in the next slide. Regarding bovine and porcine, and this has been spiked in CHO cells. Regarding bovine and porcine, a pool of 25 homologous synthetic RNAs in BT cells. Regarding human and simian viruses, a pool of 29 synthetic RNA spiked in 293 cells. So the cell pellet lies in RNA extraction kit were spiked at different concentration. You will see by these synthetic RNAs. Control samples. So we have two types of control samples. A negative control, which is a MRC5 cell, which is devoid of any virus infection, and a positive control, which is a mix of B95-8 cells that are chronically infected by EBV, diluted 10 to -4, into Ramos cells, which are chronically infected by murine leukemia virus. So it's a mix of two viruses, one high expression, the other one low expression because of the dilution. And we have spike-in controls, meaning that in each sample, including the control and each test sample, we spike 7 different RNA, ERCC RNAs, and the coverage of this RNA should be more than 80% for 4 of the 7 ERCC targets compared to the spike in the positive control. That's derived indeed from the validation. Distant strain. So how do we mimic the distant strain? So I give you an example regarding murine rotavirus. So MAP test should detect murine rotavirus infection. Murine rotavirus is not a virus, meaning it's not a virus. The viral species is rotavirus A. There are 9 different rotavirus species. The murine rotavirus is a rotavirus A. And the ICTV, the Committee of Nomenclature for Viruses, says that the limit between two species is 53% identity at the amino acid level for the VP6 protein. So we have defined synthetic RNA corresponding to the 53% identity at the amino acid level, and it is our distant RNA. And we have done exactly the same for all rodent viruses, So method very rapidly, cell lysate, spike with a different pool, RNA extraction, spiking with ERCC, library, and nucleotide sequencing at a depth of at least 65 million reads per sample. A targeted viral pipeline for the RCC internal control and for the coverage of the targets, and indeed the agnostic analysis for evaluation of the detection. The diagnostic pipeline is as follows. It has been published. There is quality steps. Indeed, there is an assembly steps. At the end of the assembly steps, we get contigs and singleton. The contigs and singleton are translated compared to the viral protein database, which correspond to RVDB translated to protein. And in case of positive hits, we counter-check with the comprehensive protein database from the NCBI. And in case of positive hits, we counter-check once again the comprehensive nucleotide database from the NCBI. And each remaining hit is reviewed by an expert and reported. So these are the results, beginning by the rodents close and distant strains. The list of viruses here, the concentration. So at 0.1 RNA copy per cell, we were able to detect all close and distant strain. At 0.01, the same. And at 0.01, the rate of detection was indeed lower. And not only we detected the sequence, but the coverage was very good. In fact, you have here the coverage of each transcript, and the lowest one is something here 40% coverage of the transcript at the concentration of 0.1 copy per cell. Regarding now the distance strains, here the coverage is very similar. It's very high, and it's 75% coverage at a minimum. Regarding the human simian panels, all were detected at 0.1 RNA copy per cell, and it's what lower at a lower concentration. And once again, the coverage was high, and the lower one was 25% here. And regarding bovine and porcine viruses, 1 copy per cell, 100% detection, and 0.1 copy per cell, all were detected except one, but it was a bug of the report because, in fact, the coverage was very good for all these viruses, including the one that was not reported by the agnostic pipeline. So at this step, if I try to summarize, the limit of detection of this agnostic NGS test is estimated to be 0.1 viral RNA copy per cell. It detects 100% of the 78 viruses, human, simian, rodent, bovine, and porcine. And this limit of detection seems valid also for distance strains that align with the definition of this virus species from the ICTV, and this gives good insurance of reliability. So based on that, how can we discuss the predictive negative value of the result in case of negative results, indeed? So first, regarding the testing of cell banks, to do that, we have done a bibliographic analysis, which is not comprehensive, which could be completed indeed, but which give you an idea. You have a list of different viruses here, family viruses, DNA, negative RNA, positive RNA, and the viral load per infected cell, which is between 2 and 5 during active infection. And we try to compare this kind of technology with infectivity assay. So we are interested in active infection. And to give you an example, for herpes viruses, during active infection, the load is 10 to the 4, but during latency, it is nothing, or it is very low, between 1 or 2. So now, if you keep in mind that our worst case limit of detection was 0.1 RNA copy per cell, if the worst case here is 10 to 2 RNA copy per infected cell, this equates a limit of detection of 1 infected cell in 1,000 non-infected cells. And it's perfectly online with the results we have obtained experimentally in this paper last year, when, depending on the virus and on the cell line, our limit of detection was between 10 to -3 to 10 to -7 infected cells in non-infected cells. So we think that this level of analytical sensitivity obtained by an agnostic technique is sufficient to cover the risk for characterization of master cell banks and late cell banks. Second application now, discussion about testing of the bulk of recombinant proteins produced in eukaryotic cells. So I have to remind you something which is obvious, but I know that this needs sometimes to be said again. That is, eukaryotic cell culture, unlike bacterial culture or chemical synthesis processes, can support the replication and amplification of eukaryotic viruses. And this is the reason why regulatory guidelines ask for virus control specifically for the bulk derived from eukaryotic cells. And clearly, it's not the case for bacterial or chemically synthesized process, despite the risk of carryover of viruses from manufacturer, despite the fact that for bacterial process, for example, we can use very high concentration of peptones during the manufacturing process, not only for the banks, during the manufacturing process. So this risk is considered to be, of carryover, to be very low. And the fact is that we have to consider the risk of amplification by eukaryotic cells. So regarding now the effective testing NGS strategies to detect this viral amplification that could have occurred during cell culture, there are two possibilities. The first one is a viromic analysis on both intact or disrupted cells and supernatant, indeed, as viral particles can be found associated to the cells in the supernatant, in both. So you have to test both compartments, DNA, RNA, etc. And this dual target approach, we think, may dilute sensitivity compared to the transcriptomic approach. And in addition, when you use upstream of the process inactivated, for example, bovine serum, you will find nucleic acid and it gives you a signal. But using a genomic approach, you cannot make the difference with something which is replicating. So we prefer using a transcriptomic approach when cells indeed are available at the end of the fermentation process, live cells, which is generally the case for biotech product. I'm not speaking about vaccines, for example, or viral vectors, but biotech product, generally, we have live cells available, and we advocate for the use of this agnostic transcriptomic analysis. And third application, which is very interesting, I think, very specific, very particular, it's testing of CAR T cells for herpes virus 6 and 7 infection. So why? Because a very high proportion of adults are chronically infected with herpes virus 6. The rate of detection in donors is between 9 and 90%. And this corresponds to DNA. So you cannot conduct testing by PCR, DNA PCR, because you will reject a number of batches of donors. It's not possible. So these virus genomes are episomal or integrated in a fraction of the body cells. And in addition, in 1% of the population, there is an integration in the germinal cell line. So the genome is passed to the offspring, to the baby, to the future donors. So 1%. But in both populations here, the virus is latent, so no risk. But, and this is a publication last year in Nature, a very interesting publication, in vitro, during the differentiation of CAR T cell, a subset, a teeny subset of CAR T cells reactivate, and this is associated with the surexpression of the virus receptor. So reactivate, and this corresponds to between 1 in 300 to 10,000 cells. One reactivating cells in 300 to 10,000 cells. So on virological aspects, during latency, there is no RNA secrete, there is no transcription at all. This is perfectly demonstrated by using PBMC from patients without viremia. This is demonstrated using lymphoid cell lines with latent herpes virus infection. And this is also well demonstrated using PBMC from these ICI HHV6 patients. All PBMC harbor DNA, which is latent and no transcript. This means that it's not a question of level of detection. There is no transcription. And this contrasts clearly with active infection, where there is a wide range of transcript, the main ones being early genes and more particularly the polymerase gene. So our proposal regarding the strategy for testing is to use RNA-seq, so transcriptomic, on cells that are cultivated, in fact, 2 weeks post-infusion time, typically 3 to 4 weeks after the beginning of the differentiation, because it has been well demonstrated that the rate of reactivation increase with the differentiation time, and this mimics the risk of reactivation after infusion in vivo. So use this RNA-seq approach with an acceptance criterion, which is no detectable HHV6 or 7 reads after an extended culture time. And the reliability, the predictive negative value of this test indeed depends on the limit of detection, of the RNA-seq used. So if you consider that our limit of detection is between 1,000 in general to 1 in 1,000 in general to 1 in 10,000, that seems to correspond to herpes viruses non-infected cells. This seems satisfactory compared to the ratios of cells that reactivate the virus. I have said 1 in 300 to 1 in 10,000. So take-home message. So the limit of detection from non-regulated R&D testing of this GMP transcriptomic assay technology is estimated to be 0.1 viral RNA copy per cell, or during active infection, less than 1 infected cell diluted in 1,000 non-infected cells. It detects all viruses, including distant strain across these human, simian, rodent, bovine, and porcine viruses. And there is an application possible to cell banks, indeed, but also cell therapy, drug product, and bulk protein testing of recombinant proteins. And it's a pleasure to thank the team of PathoQuest who participated to the work and underline the participation and collaboration of Charles Rivers Laboratory. Thank you.
All right. Good afternoon, everybody. Good evening, good morning to people online. I'd like to thank the organizers, first of all, for accepting this presentation. I'm going to be talking a little bit about the matrix effects of limits of detection for NGS based on some of the adventitious virus detection assays. Sorry, just trying to, there we go. All right. So first of all, I just want to say, like, we're a CRO, and so there's a couple of unique advantage points of being a CRO. So first of all, we work with multiple clients across a cross section of the industry, testing a lot of different modalities. So we're testing viral and gene therapy products, we're testing CHO products, we're testing vaccines, and we see a lot of different products from a lot of different clients. We also have experience addressing regulatory feedback, and this is true of all CROs from multiple applications of the technology, specifically here for NGS. So we've seen feedback from the regulators, we've seen what they're requesting, and we're working with them to try to get them the first time the information that they need to properly evaluate a product. We also have multiple validated methods for different purposes. So we have the transcriptomic versus viromic versus genomic approaches. Another advantage is that we do have NGS experts. So I have a whole department full of NGS experts that do that, as do other CROs offering next generation sequencing services. And what I really want to focus on today is we have an accumulation of data in multiple products and backgrounds through the use of spiking studies. So as you've seen, the spiking studies are performed to provide that additional layer of limit of detection and validation for your specific products. As a CRO, we don't see one product or two products, we see multiple products. And so I'm going to be able to share some of that information, anonymized, of course, in the presentation a little bit later. And just to focus on the pictogram on the right, these are all fully validated GMP methods, validated fit-for-purpose analysis algorithms, data integrity, secure data and storage and transfer, matrix suitability, and high-quality sample processing. Now, in terms of validation, we've all heard a little bit about this, so I won't spend too much time here, but NGS is validated in a modular fashion with extraction, library preparation, sequencing, and bioinformatics. The approach confirms that each portion of the assay behaves as expected, and it offers a lot of flexibility for updating rapidly changing technologies. So you can, in theory, update the library preparation or the sequencing without impacting your other modules. Now, limits of detection for client-specific matrices are really established through the end-to-end analysis using known viruses to achieve your ICHQ2 parameters. So those are specificity, those are conveyed by the algorithm, confirmed in validation, and limit of detection is really established on a matrix-specific basis. And then your full validation package really consists of your generic validation plus any additional spiking studies in your specific products. And you can see the NGS method sort of over here. It's very much the same for everything. We discussed this yesterday. You pretty much have your test sample material. You got your nucleic acid extraction, library preparation, sequencing, and data analysis. And then, like Peju was going over yesterday, we sort of have this bioinformatic approach where we have an algorithm. We identify potential positive samples. We then screen those against the BLAST database to determine if it's a legitimate hit. So in terms of spiking studies, just moving on, we do, this is just a general overview, we do use the representative reference viruses that are available through BEI resources from Arifa Khan's efforts. The viruses are qualified by worldwide laboratories and are standardized to be used in NGS AAT applications. So we have the old 5 viruses, and then we just have the 2 new ones that have actually become available more recently. And the model viruses really are meant to represent different virus qualities and worst case scenarios. So they do contain different physiochemical properties in terms of DNA, RNA, strandedness, segmented, circular, envelope, non-enveloped, chemical resistance, et cetera. So they're meant to be truly representative of what you could potentially find in products. And so what does a spiking study need to look like? I think we went over this a little bit already, but we sort of have all of our viruses here. Most of the work that we have done so far is with the 5 viruses because the 7 viruses have just become available. And so you've got your 7 viruses, you spike them in at different concentrations into your test matrix, and then you perform your bioinformatics. A couple of general guidelines that we follow, we do use our modular validated methodology. We use the client-specific matrix as representative material. In general, we actually perform a pre-study before we go into it, where we spike samples using a high concentration 1E6 to look at the numbers and see where we think the most likely limit of detection for a particular matrix would be. So that way, we're not jumping directly into a spiking study without any knowledge of where we might actually end up. That way, we can focus the spiking study a little bit more. Depending on the applications, it was 3 to 5 representative viruses if you're looking at RNA or the RNA and the DNA viruses, and a number of concentrations, and of course, replicates at each concentration. When we do the data analysis, we're using the non-targeted approach. We're essentially running this through our agnostic pipeline. Even though we know what viruses we're looking for, we're making sure we can detect those viruses with the same process we're going to use to analyze the sample to look for unknown viruses. And with that, we have our criteria, because these are all GMP assays in terms of specificity, homology, fragment length, and number of reads to be able to call something a potential positive result. Again, we've got those replicates, and we've got required depth of coverage. So we'll actually use the spiking studies and look at the number of hits in terms of hits per million in order to determine how many reads do we actually need for that to hold up in routine testing. Do we need 100 million? Do we need 200 million? And what if we go below that, then the limits might not hold up. So in terms of general performance of spiking studies, I showed a similar graph of this at the PDA conference, actually updated it with year-to-date information. We've seen a pretty significant uptick after the finalization of ICH-Q5A at the end of 2023, the new draft chapter coming out in 2024. So far, year to date, we've seen pretty much double the number of spiking studies that we've seen in previous years. And you can see the uptick here. It is company data, so I had to blind the access just so that we're not sharing all that information. But this is the general trend that we do see. And we do see samples, bulk harvest, we see master virus seed, we see master cell banks for these limit of detection studies. Now, in terms of sample preparation, we've heard a little bit about the different methods. We have this transcriptomic assessment where we're looking at the RNA in the cell, and we have the viromic and genomic assessment where we're looking at more total nucleic acids for the genomic and then encapsulated nucleic acids for the viromic assessment. Now, really, no matter what method you perform here, we use Illumina-based technology for all of our NGS applications currently. You come to double-stranded DNA library preparation and then downstream sequencing. So in terms of cell lines, what we did here, we actually published on this, and my colleague Rebecca actually has a poster next door if you're interested in learning more. I'm not going to go over the study in a whole lot of detail, but we did take representative cell lines in the industry. This is more for your cell line validation qualification. We took CHO, human viro, SF9, so 4 representative cell lines. We spiked them with 3 RNA viruses from the panel to look at what the limits of detection for those 3 viruses were. And the information that we found is it's represented here on this axis. You can see the viral genome copies per 1E6 cells. And so we did see limits of detection of RSV, FELV down in the 1E3 range. For most, Rio was up in the 1E4 range, which is pretty consistent with what we've seen with reduced limit of detection of the Rio due to its double-stranded RNA nature. And basically, what our data showed is for all of these, because we set our limit of detection as the worst possible of all the viruses, because we're not looking to detect all these 3, we're looking to detect any virus, as 1E4 virus genome copies per 1E6 cells are essentially 1 in 100. Now, if a client comes to us, like these are the 4 most commonly used cell lines in the industry. If a client comes to us with a significantly different cell line or highly modified cell line, even of the same nature, what we'll do is we'll do this LOD verification in client-specific cell lines to confirm that limited detection is consistent with what we saw in the validation. And actually, in all cases to date, because we've ran the gamut here in terms of CHO, human viro, and an insect cell line, we're actually able to confirm that limit of detection in cell lines. So cell lines are pretty predictable for us. Now, in terms of non-cell-based materials, this is going to be your bulk harvest, your master virus seed, your virus-based materials. They have a matrix that you have to contend with. And what is the matrix really made of? It's made of the enzymes, the buffers, the solvents used in sample processing. It's made typically of a recombinant virus in the case of a virus-based product. And these are all viruses that could potentially be there. And as a CRO, again, another advantage of the CRO is that we've actually tested all of these viruses in some way, shape, or form in a matrix that our client has provided to us. And I'm going to show a little bit of data on that in subsequent slides. Also in your matrix, you've got cells, you've got host cell nucleic acids, and that can add to the complexity of your matrix. Now, a couple of things to remember for NGS, it is an agnostic analysis, so you are looking for everything in there. Generally with NGS, and I say generally because there are amplification methods to use if you're looking to target specific viruses, but in general, there's no exponential amplification for the agnostic approach. So essentially, the background of your nucleic acid content matters greatly in terms of concentration and limit of detection. And we've seen that if you have a highly concentrated virus or if you have a lot of residual host cell material, you end up reducing your limit of detection because you have so much other nucleic acids in there that is sort of sucking up all of the reads there. Now, the WHO endorsed NGS reference materials, just another thing to keep in mind, were qualified in adenovirus background only. That was 1E9 viral genome copies per mil. And then the question becomes, is this a truly representative matrix that we can extrapolate to multiple viruses? And I'm going to show data on the next slide. I promise I'm getting there. That's going to that's going to say that it's a little bit different, generally predictable, but a little bit different in different matrices. Now, some specific challenges here are host cell nucleic acids, the presence and the concentration, some potential solutions we've seen presented in the guidelines. One of them that we use is we use a nucleic acid degradation like benzonase to remove those host cell nucleic acids. Another specific challenge is viral nucleic acids. Not only the presence of a virus, but also the genome length and concentration. If you've got a really large virus, and we've seen this before, that's very highly concentrated, if you're in the 1E10, 1E11, 1E12 range, that virus ends up sucking up all of your reads. And so your limits of detection become less and less, but you can't get rid of that with benzonase. And so because it's an encapsulated virus. So now we're looking at potential viral nucleic acid depletion steps for that, to solve that problem. Now, this is the data that I promised that I wanted to get to. I'll walk you through this real quick. And so we have a lot of different viruses that we've tested in different backgrounds. And these are all different viruses. I had to anonymize this data because it could be identifiable to particular clients. But being a CRO, we sort of have this repository of all of this data. This up here is our biggest one in a Vero background. And these are all different samples. So we have 7 different samples that we've run in a Vero background that we've performed spiking studies on. This isn't everything, but it's a selection of what we've done. So all the way down on the left side here, this one was actually COVID in a Vero background. We actually see limits of detection of all the virus spikes at 1E3, which is actually one of the best that we've ever seen. If you think about COVID, it's an RNA vaccine, generally lower concentration when you're manufacturing in Vero. Now, when you get up here, you see limits of detection suffer a little bit more. You're at 1E6, essentially, for Rio and PCV, and then a little bit better for EBV, the more hardy viruses, FBLV and RSV. And this was actually a pox virus of high concentration. And so that's what we see there. And now all of these are sort of, they're different preps, they're different viruses, different levels of residual DNA, just trying to illustrate here that the matrix actually does have an impact. Although we do generally see limits of detection fall between this 1E3 to 1E6 range for pretty much all of those viruses. Then we move to the human background here. It's interesting because these are actually adenoviruses of different concentrations. And so the adenovirus that we qualified, the reference standard was a 1E9. These are actually higher concentrations and had a lot of human residual DNA in there as well. And so we see higher limits of detection in this particular sample set. Then we move to the avian background here. Now, avian allantoic fluid, pretty clean in terms of residual nucleic acid. Your influenza vaccines are typically made in this, so not large particular viruses. And this is where we see Rio and PCV in the 1E4 range, and the rest of them down into the 1E3 range. This is pretty characteristic of what we see for this type of work. And then this is a pain point for us right now. We've got an insect background here. Most common virus in insect line is a baculovirus. So this is very high concentration baculovirus. And when we run it, even with a benzonase treatment, this is actually with a benzonase treatment, we see the limits of detection suffer pretty much for this. So what does this all say? It all says that we've used the same generic method. The method that we used was actually used to help qualify the reference material. So it's actually comparable with all the other labs around the world because we did that collaborative study. But it shows us that that one method might not be suitable for every single matrix when you're trying to get all the way down. But then the question becomes is how low do you actually need to go? And so that could be based on a risk assessment of your product. What filtration steps do you have later down the line? And we discussed that all in some previous talks before. Now, I did want to share one case study here, the effects of benzonase, because we say we use benzonase. Does it have a profound effect? Does it not? So I picked the most extreme example here. And I'll just give a little bit of context here. So in the US, we source fetal bovine serum, FBS, to use in our in vitro-based assays. But there's been an issue with contamination of that fetal bovine serum with influenza, bird flu H5N1. There's been some literature early in the year. There were 93 flocks infected. Now it's more in the hundreds. And so what we've decided to do as an organization is screen new lots of FBS using next generation sequencing. And so in order to do that, each lot has its own characteristics. So we're actually spiking in the viruses every time, both with and without benzonase here. And so for this particular experiment, we spiked in the viruses at a high concentration, 1E6, to see if we can detect them. And as you can see here, this axis is actually incorrect. Sorry, I realized this late last night. It's actually number of reads per million. It's not VGC per mil. And so what you can end up seeing here is that when you test with benzonase, benzonase is in the purple here, you see pretty high copy numbers. But when you use no benzonase, again, FBS, it's got a lot of residuals in there. It's got a lot of viruses. It's got a lot of stuff. All the reads are essentially sucked up by that particular background. So in this case, we were able to reduce that background to visualize some pretty good limits of detection for the 5 representative viruses. And so, in summary, we are a CRO. From a CRO perspective, what we've seen is we've seen cell lines behave relatively consistently in terms of limit of detection using representative viruses. For complex matrices like your bulk harvest, master virus seeds, they may show differences in limit of detection depending on the matrix composition. And you may actually require some matrix-specific processing steps along the way. Key variables that we see seem to be recombinant virus type, concentration, virus size. And this is where, as a CRO, this is my last plug for CROs, I promise, but this is where we have a lot of prior knowledge and can help guide clients in terms of what we've seen for those particular products and how we can most optimize your limits of detection. One representative complex matrix that we've found doesn't currently exist. There is a good model out there with an adenovirus, but it does need some product-specific considerations when we're testing client-specific matrices. And then spiking studies using the matrix-specific representative material will likely continue to be performed, although they can likely be streamlined through limit of detection procedures. So because of that prior knowledge, we can take a best guess, or we can do a pre-study to see where we think limits of detection are going to be. We can also evaluate the product to see what we actually need to achieve and then verify that limit of detection. It'll help streamline the process a little bit. It'll help reduce costs so that you don't have to perform a full spiking study every time. And then, of course, the key questions out there: is there a minimum acceptable LOD? And to some effect, this really needs to be part of your risk assessment, the downstream processing, and that kind of stuff, taking also into account what a true infection would look like at your point of testing, which is likely addressed on more of a product-by-product basis. So I'd like to acknowledge a couple of people, Layla, Mackenzie, Allison, and the global NGS team. And then we do have a couple of posters. Rebecca and Amber are both here, and they have some posters next door if you guys are interested in learning a little bit more. All right, thank you for your attention.
Thank you for the kind introduction. Today, I would like to give you an overview about the spiking study 4 that aim to evaluate the longer read sequencing for the adventitious viral detection in a high titer viral background. The study is led by Simone Olgiati and myself. So first of all, the disclaimer: the content and the view expressed in this presentation are those of the individual expert participating to the spiking study 4 and should not be construed to represent the view or policy of the organization, regulatory authorities, or governmental agency they represent. All criteria, tool, or way of working described must be considered as consideration only and should not be taken as recommendation, mandatory views, or regulatory views. So, boring part finished. So, I will go briefly to introduce you the working group, as was already mentioned in a previous presentation. So the mission of the working group is to facilitate the implementation of new technology, such as NGS, by providing informal scientific forum, mutual exchange among different types of organizations. And the way that the working group would like to reach this goal is to provide feedback in different subgroups that address different NGS topics, both on the laboratory part as well as on the bioinformatic part. In parallel to this, there are some collaborative studies that are based both on short read technologies and long read. And today I would like to give you an overview about the spiking study 4 that was the first spiking study based on this type of technology. The purpose of this study was to evaluate the long read technology with a viromic approach to mimic a potential adventitious contamination in a viral seed. And to do that we select as a background model, the adenovirus 5 that was spiked with 5 selected virus that belong to the CBER NGS virus reagent that has different genome types and different physical or chemistry feature. In the study are involved 12 participants, and we expect to complete the study next year. We select as long sequencing technology the nanopore 1, and briefly for those who are not familiar with the technology, the nucleic acid is guided through a pore and passes through a membrane. Into the membrane is applied an electric field, and the difference of charge of each basis allow to proceed with the sequencing reaction. At the time when we started the study, we have a limited experience on the NGS, on this type of technology. Therefore, we thought to have benefit for the application of viral detection. And so we decided to evaluate the sensitivity and the specificity, as potentially with one long read, we can identify the viral genome that we want to detect. On the other hand, we know that there were some issues about the technology or some constraints, for example, the error rate. And so due to this fact, we sought to start with a simple matrix. So we use a viral seed background, as was done for the Illumina based study that was based on a short read. The plan at that time was to proceed with other metrics background, more complex, such as a cell background or unprocessed biocarvers, or try different approach. But discussing within the group, the collaborative group, and with the working group leader, we decided last year to split this part and to move to other collaborative study, like the spiking study 5 and 6. So I'm talking about the experimental design of the spiking study. The starting material is composed of the adenovirus at the 10 to the 9 genome copies concentration in 200 microliter. And this background was spiked with all the selected virus. All the participants have to test 2 spike level, the 10 to the 4 and 10 to the 6 genome copies. They were allowed to proceed with the study with a common protocol or an individual protocol, and to apply a target and a non-target approach. So in the bottom part of the slide, you can see the timeline of our study. We start to discuss the study in 2019 at the YARBS conference in Ghent. So face-to-face meetings are a good opportunity to new idea, as well as coffee break, as mentioned this morning. We start in 2020 with a survey to evaluate what were the expectation of the participant, what mature was the technology within the group, and what type of sample we want to address. It took some time due, first of all, to the COVID period, and then to sign the CDA, to design the experimental part, and obtain the common starting material. This year, we were able to collect all the preliminary data for the target results, and the way that the collaborative study work for collecting data is a common collection sheet that in this case was adapted and adjusted for the longer read technology. To gain and to explore this technology, within the group was shared a common protocol. As you can see, it is composed of different steps that are the classical steps of an NGS, but with a focus on longer read. So first of all, the sample preparation. So we use a common protocol to prepare viral stock dilution and prepare spike sample. Then we go through the extraction phase to recover both RNA and DNA. Of course, a reverse transcription step because we have some RNA viruses. And then through the library prep and sequencing and data analysis, which was individual for each participant. Some points to consider into this workflow. First of all, the low quantity of nucleic acid. As we are talking about viruses, the content of nucleic acid is very low. And moreover, we have to preserve the long fragment as we are dealing with long reads. Another point to take into consideration is the chemistry and the software update. As the technology is evolving very fast, we had to consider a different chemistry for the library preparation, as well as different version of the sequencing instrument. So with that, I conclude my introduction part, and I'm going to show you some preliminary results. And within the group we discussed, and we sought to divide this part in two. First of all, I would like to give you a flavor of the sequencing metrics of this technology. And the next three slides are refer to the three common samples that were analyzed from all the labs. So the AD5 background and the 2 spike level 10 to the 4 and 10 to the 6 genome copies. The first slide refers to the total number of reads. So as you can see, the range is between 8 KB and 40 million of reads. There is a sort of variability among the different samples and among the different labs. It has to be noticed that the highest number of reads was observed for the spiking level, the higher spiking level in most or in the majority of the lab. An interesting data could be the mean read length, as we are talking of long read. So what we have observed is that there is a range of variability that go from 500 bases to 3,000 of bases. And against, for this parameter, you can observe a different level of variability among the sample and among the different labs. Interesting to see that in this case, for the higher spike level, we observe the lowest mean read length. And to talk about the quality, this slide refers to the mean read quality that was observed in the three samples, the three common samples. So you can see that the quality is between the Q7 and the Q17, and each laboratory obtained a consistent Q score among the different rank. However, also in this case, there will be a sort of variability among the different labs. Now I'm moving to give you a preliminary overview about the sensitivity of the technology. So in this part, you can see of a table that report the percentage of laboratory that could able to detect the five viruses. In pink are highlighted the three common samples, but each lab was free to test a different spike level to assess how was the sensitivity of this technology. And as you can see, all the labs were able to detect all the viruses at the 10 to the 6 genome copies, and most of the labs also to the 10 to the 5. This table is referred to the previous data set. And so we would like to show you the number of mapping reads detected for each type of viruses, considering the package of the data available. And as you can see, there is also in this case variability among the data at the different spiking level. And these data refer to the target analysis. So, to conclude this presentation, we have seen that all the viruses were detected at 10 to the 6 viral genome copies. And we would like to highlight that the technology is also able to detect circular virus, even if the sensitivity was not the same for all the labs for this type of virus. We have observed that high spike levels displayed higher number of total reads with lower mean lengths, and this is due probably to the fact that the workflow was set up to detect viruses, and in the higher spike level, there is the majority of viral spike. And so in this case, we have short read length. For the next step, we would like to aggregate the non-target analysis results. And of course, when all the data package will be available, we will make some consideration about the individual protocol or the common protocol if there are differences among the results that we obtained. This is my last slide, and we would like to share some lessons learned during this study. So first of all, the technology is rapidly evolving. So there are frequent updates about chemistry, about software, and this is a point to be considered in case we want to compare the results. Some points about the workflow. So we need to evaluate the starting material, meaning the quantity and the quality of your starting material to prepare and to process your sample. And of course, you need to preserve the long nucleic acid. For the sequencing, we have observed some variability in flow cell from batch to batch. And another point that we would like to address is the quality criteria for base calling. And we would like to evaluate if there is an impact on sensitivity. Regarding the analysis, we have observed that the presence of full-length reads that were longer than the expected viral genome size, and this was due to probably a chemistry artifact of the technology. And finally, you have to consider that when you are analyzing long read, you have to adapt, of course, your pipeline. These are I would like to acknowledge all the spiking study for member. And as you can see, the group is large and include different type of organization, regulatory agency, company, and university. And so with that, I conclude my presentation. Thank you.
Reported, well recognized. That is really performed without any prior sequence information. So what were the goals of the project that we developed? And this has been going on for a few years now and is now completed. The goal of the project was to obtain specific NGS data that allowed us to make decisions regarding the suitability of different applications for NGS for adventitious virus detection in biologics products. So the first question we wanted to address was NGS able to replace animal-based adventitious virus testing? Were we able to develop tools in terms of support for in vitro adventitious virus testing and identify new in vitro release tests to replace animal-based testing for vaccine manufacturing quality control? So the key aims really were to evaluate next-generation sequencing as a virus detection method in a complex biologics material, in the background of that material, and directly compare that with the traditional broad spectrum in vitro and in vivo assays, using the same spike samples, virus samples, for testing in all of the different viruses or different assays, my apologies. We also, as second part of the project, was also to look at the enhancement of NGS bioinformatics for known and novel adventitious virus detection. That was by refining the reference virus database to increase the specificity and accuracy, and to look at how the pipelines may be made automated and efficiency of the NGS bioinformatics work. And Pérugen will actually provide us with that information in the next talk, so that it will not be part of this particular presentation. So there were a large number of people involved in the project, and it was a multi-group study. So we had project teams from various different groups. From the US FDA CBER team, Arifa Khan led the project, and her team were heavily involved. Merck and Millipore are the same company, just to avoid any confusion. So we're Merck here in Europe, Millipore Sigma in US. We have a number of different teams from the Merck organization involved in this group. So effectively, we had the contract research organization BioReliance involved, as well as our Merck colleagues from healthcare. We also had a team from the GSK group, and the work that was performed here was very much wet lab work, and some of the bioinformatics work was also performed here. And as I've mentioned, the talk that we will have next will cover more of the bioinformatics enhanced workflow that was performed both by the US FDA and also by the University of Delaware. As we've mentioned, the project is funded by a grant from the National Institute of Innovation and Manufacturing Biopharmaceuticals, NIMBLE, under PC 3.1305. I should know this off by heart. The study was set into numbers of different parameters. So you'll see on the left-hand side the organizations that were involved and the different steps that were performed and the groups that were involved with that particular work process. So there was obviously specific viruses and matrices that needed to be prepared, tested and characterized before you use those materials. And that was basically work performed by both the FDA and by Merck. Samples were prepared, spiked samples were prepared, and that was solely prepared by the team at the FDA. Pre-study analysis was performed also to really characterize some of the materials that had not had a specific level of characterization performed. And also pre-studies were also performed to determine the suitability of the in vitro assay in particular is in association with any assay interference that may be impacted in terms of the testing. Remember, we're running traditional methods as well as the NGS system. The NGS was performed by all three groups that were involved in the study, and 2 laboratories performed the in vitro assay and 1 group performed the in vivo assay. So the study considerations. Obviously, in italics, you'll see here that it's presumed that NGS is a superior method to in vivo and in vitro assays. There have been a number of different publications looking at that aspect, but not looking at a true head-to-head comparison of these tests. And it was really to look at the breadth of virus characterization and NGS and looking at the sensitivity of the test and showing that there is a level of similarity in terms of virus detection in NGS versus the PCR methods. The method of the study was designed specifically to challenge the NGS workflow and to look at the detection of the biological assays that were selected and look at the selected viruses that were known to be at minimum infectious in at least one of the target cell lines in the in vitro assay or one animal species in the in vivo test. So to achieve the goal, there was a very large amount of time spent looking and reviewing literature and ensuring that we had the right selection of virus. We chose 2 of the 5 CBER NGS virus reference reagents for adventitious virus detection. These were used. The breadth of NGS, unfortunately, could not be evaluated because we weren't really comparing these to the specific requirements of the other assays in the study. We only were testing really the 2 viruses. So we appreciated that that was not basically something we could challenge, certainly in this study. The 2 RNA viruses used in the study were human respiratory syncytial virus, a single-stranded enveloped virus, and Rio type 1, a double-stranded non-enveloped virus. And based on comprehensive literature and of the ABTIC studies, in particular study 2B, we also know and we're aware that the NGS can detect viruses that may not or would not necessarily be picked up by the classical standard in vivo assays or in vitro tests. So a little bit more detail on the study design and the sample preparation. So as I've mentioned, the viruses that were selected were human RSV and the Rio virus. And they were selected due to the fact that they had been already well characterized as part of the virus reference reagents. They were characterized with various different analyses. Many of you have already heard those analyses that were performed in some of the talks earlier today. And we also looked at really evaluating in-house digital droplet PCR testing, which is available in the FDA labs, again, as a basic supportive data for the actual viruses in question. The sample type that we chose was a very complex CHO-based cell bioreactor material. It was expressing a monoclonal antibody, and it had a high protein concentration. And again, prior to the actual initiation of the study, this matrix was actually evaluated fairly comprehensively, both for assessment of standard quality control characteristics, both for microbial detection, as well as evaluation of host cell proteins, looking at the quantity of host cell DNA. And we also were assessing that, in fact, there was no toxicity that would affect the recovery of the viruses in the presence of this background material. So a little bit more detail in terms of how we prepared the spike samples. We prepared 6 spiked samples and 1 unspiked sample. There was 12 10-fold serial dilutions performed for each virus, and they were fixed in a volume of matrix, and the samples were blinded. So the tests that were performed within the laboratories were completely blinded to the experimental teams that were working on this. These were all stored appropriately at -80 prior to distribution, and the aliquots were made as a one-time test for the experimental work. 6 samples were selected as part of the pre-study to determine the range of infectious titer for the work and also the genome copy analysis in order to ensure that we would produce a positive result in the test systems with at least one of the viruses. And for a head-to-head comparison, the same volume of material was tested by digital droplet PCR, NGS, and the in vitro assays. And the same test was also performed, well, same materials were performed in the in vivo, but we were very conscious of ethical concerns around performing an extremely extensive level of testing in the in vivo tests. And we minimized the number of animal tests that we perform and used a tiered strategy to really start with the lowest concentrations, or lowest dilutions, I should say. And then we took the test systems based on those particular results to evaluate if more testing was required. The virus tests that we performed, as I've already mentioned, and I'm sure probably most people in the audience are very familiar with these assays, but we chose a 28-day in vitro cell culture test, and we used the 3 classical cell lines that you would perform for a standard CHO cell-based material. So VeroCell, MRC5, and CHO-K1 cell lines were used. The 2 participating labs used the same experimental setup as per standard traditional methods. Appreciate we call them compendial. They are mentioned in the compendia, but they're not specifically specified in terms of the testing that's required. So the methods really were evaluated extensively in terms of the similarities for the test. We also used classic observations of cytopathic effect, heme absorption, and hemagglutination, and those were noted during the study period as applicable. The in vivo assays, again, were performed in accordance with standard regulatory procedures and were performed in suckling mice, adult mice, and embryonated hen's eggs. And the age and the number of mice and eggs and the volumes and routes of inoculation and incubation times and the readout in terms of clinical evaluation were all performed in accordance with the standard assays that are performed in industry. We also included, as is standard and is normal, to do a sub-passage for both the suckling mice and the embryonated hen's eggs. We also performed next generation sequencing, as this is a next generation sequencing meeting. This is probably the part most people are very excited and interested to hear. We used short read technologies. Each lab used their own protocols for the entire workflow, including the bioinformatics pipeline. We wanted to evaluate the differences between the teams. And to ensure results for virus detection for the bioinformatics in particular, the teams worked very closely to make sure that the same accession numbers for the reference virus sequences were used for targeted analysis, and the RVDB database was used for non-targeted analysis. And again, the digital droplet PCR test was used to release these fine results for the pre-study and to use those to determine the sensitivity of the virus detection. Let's go into some data, and I will summarize this. So I will just summarize and hopefully just navigate you through what this table actually shows. So what we have on the left-hand side is basically obviously unspiked material. We then have viral genome copies per mil, and virus infectious titer of TCID50 per mil, and a wide range of particular specific test ranges from 10 to the 7 down to about 14 copies. And 10 copies, or yeah, sorry, my apologies. Down to about 10 to the -2 in terms of the infectious units. The NGS representing the targeted bioinformatics from the 3 labs. And you will see that there is some differences in terms of the recoveries for the different labs. The positive criteria for the targeted analysis for laboratory 1 was greater than 2 reads, and greater than or equal to 1 read for labs 2 and 3. The in vitro test, again, remember we're using the test to look for the virus in different cell lines, and the positive result was based on the recovery in at least 1 cell line. And we basically were showing again the levels of recovery from two labs, labs 2 and lab 3. And then the in vivo test, and we performed both passage and sub-passage testing. There was no recovery in terms of the adult mice and the basically embryonated hen's eggs, but the suckling mice, we also in this particular virus saw no impact of that particular virus on the actual in vivo assay. The summary of the Rio virus was perhaps not wholly unexpected. Again, the different ranges on the left-hand side of the chart. We again saw some differences in terms of recovery of the specific viruses through the NGS analyses. The in vitro assay was picked up at all concentrations by both labs, and we did see some impact in terms of the in vivo assay in both the passage and the sub-passage recoveries. So in some ways, and we will go into this in a moment, the in vivo assay, if there is a virus present, it will pick up virus where it is appropriate. But there are some considerations that we need to make in terms of the recoveries that are evaluated. So the study results, essentially, as I mentioned, the next generation sequencing detected both RSV and Rio in this highly complex background material of CHO cell matrix. There was differences in sensitivity, and there was differences for the 2 viruses across the different basically labs that were involved in the study. And the differences really were associated with some of the actual protocols that were used. We looked at the differences of the NGS workflows. Some labs performed pre-treatment, some did not. Sample preparation was obviously deemed to be very potent, and also looking at any levels of differences between the different protocols. What we did see that we were showing that, in fact, there was a similar sensitivity of virus detection to the digital droplet PCR work that was performed, and we were showing a similar level of recovery in terms of the concentrations that were detected. In the in vitro assay in labs 2 and 3, the results of the in vitro cell culture assay showed again similar results for Rio. And again, that was using an independent source of cell lines, reagents, an observation of the results across the read times. So whether that be CPE, hemabsorption, or hemagglutination. And remember, the hemabsorption, hemagglutination is really included in the test to detect a specific range of viruses that are affected by hemagglutinins within the actual viral materials. Differences in the virus detection was seen within the RSV. But in general, we did detect high sensitivity of virus detection in the cell culture assays. And when we compared this with the NGS, we did see some level of impact in terms of the recovery profiles from in vitro and NGS. We did pick up a positive result in Rio. It was based on subpassage. There were clinical signs. And in terms of standard practice, we would normally have gone into very high levels of specific and investigatory work. We were aware that we were spiking virus into these particular models. And therefore, we also knew that they were under very heavy ethical control. We did do a passage, and we did do the subpassage in the suckling mice, and we did pick up the virus, and we picked up the virus very nicely. We did modify the procedures, and we will report in terms of that modification and whether that can have a significant effect on recovery of potential virus agents using this particular method. I just want to go into a little bit of detail around the in vitro tests. They were interesting, and we did separately find these to be, they basically provide us with the results we expected, but we also wanted to really just maybe evaluate these a little bit more detail. So, of course, we know that our principles of basically ensuring virus safety within the actual biologics process, very clearly defined in ICH-Q5 R2. The key methodologies that are used, and the classical, I think somebody mentioned this morning, is the gold standard, is still the in vitro infectivity assay using selected cell lines based on probably risk assessment of potential contaminants. In this study, we knew there was a CHO-based molecule. We knew what the test system would be applied, and we really applied that. And we also used appropriate endpoint analysis as we would perform in a standard culture system. The study does provide experimental data and shows that there are levels of comparability of detection between the labs, probably likely based on the choice of cell line, the pre-treatment of the materials, and also just the analysis of the endpoint tests. The similarities between the labs was quite clear. Many years ago, I did a presentation on in vitro testing, and there was a discussion as to whether we should standardize in vitro assays purely because of exactly this. There was a lot of variability being detected and shown within the test, within industry. And I think this also highlights exactly that, and it will be published data that we will provide. The RSV and Rio tests were detected in replicate samples, primarily by CPE. And really, we were really detecting a positive result as really a recovery in any one of these endpoint analyses. The positive and negative controls that were used in the study came up as positive as we expected, or negative as expected. And both viruses were detected by CPE, but only Rio was detected by hemagglutination testing using Rhesus monkey red blood cells at the 2 temperatures that were defined. So the study does show that there are some limitations in terms of the viruses, in terms of your sensitivity levels, depending on how you actually perform those tests and the differences that are used in terms of different selections of test system. And it then provides a level of evidence that supplementary or replacement of the assay using NGS should indeed be considered in terms of covering the wide range of detection of assay. Similarly, the NGS study outcomes. This is really the first study that has performed direct comparison of NGS and the traditional in vivo and in vitro assays using the same sample matrix in all of the tests. And the results do go some way to actually recommend the use of NGS in the updated guideline, the ICH-Q5A guideline. In particular, in terms of replacement of the assay, we know that NGS can provide defined sensitivity and breadth of detection, and it very much aligns to the 3 Rs principles where we're trying to reduce animals for testing. The PCR assays, again, NGS was shown to have a similar sensitivity to the PCR assays and has that broader range of virus detection that we expect. And certainly the supplemental or replacement assays, the in vitro assays, in particular looking at cell substrate analysis, they're basically, if there may be some concerns over the particular recoveries of virus, you may use this test. But I think the data that we've just shown also shows the variability of the assay and the requirements to really have supplementary assays. We never run one test in biologic safety. We run multiple tests, and it's for very good reason. We want to make sure we cover the breadth of detection of virus that is required. Certainly, the use of NGS, as Arifa has mentioned earlier, is really being used to assess perhaps interference levels in some materials, and we've used that for quite a large number of tests. And also if you're using neutralized, you do not have an effective neutralization of a live virus material, you will require to use an alternate system. NGS may be used. But the study we have here does provide a really good level of head-to-head supportive data to the suitability of NGS in biologics. And we are due to submit the paper in the next few weeks to actually ensure that this is in public domain and you may read the full data analysis. I probably haven't done it justice today. There's a lot more data behind this and it should be very effective for the industry. There is more work being performed. We're actually on to study 2 of the project, and we'll be looking at optimizing the NGS for broad virus detection. We mentioned that we did not really manage to cover that part of the actual program in the first study. And we will again use complex matrices and spiking studies. And we'll challenge with the specific viruses. Again, they're well-characterized materials. And we'll also look at the well-characterized cell lines that are in Sandra's poster next door. So please do go and have a look at that really nice poster explaining the virus-infected cell lines. Also, we'll optimize the NGS bioinformatics to enhance the databases and look at the accuracy of data analysis for the virus detection systems. And we're hoping ultimately from this study to start preparing protocols, strategies, data sets to establish the technology more broadly for a more routine use. There's been many discussions today, and I think having maybe a more defined pathway in terms of technology and what is expected may hopefully help industry in terms of greater acceptance of the test system. So thank you for your attention, and I will take questions at the end.
We have heard lots about RVDB in yesterday's workshop, and also in morning and afternoon session, but you think, why is that? So RVDB is a reference virus database. And my talk today, I hopefully can convince you or make you more comfortable about using RVDB and implement it in your bioinformatics pipeline, because we do spend lots of effort, no matter from myself, from Arifa or my colleagues. So we do take lots of effort on that. So I will outline today's talk like, why do we need RVDB, since there are already many choices in public. And then I will describe a little bit about the workflow to make the RVDB. And then I will go through some challenges, especially for past few years, about the refinement of RVDB. And I will spend the rest of my talk to talk about we implement the machine learning to help us to further improve the production of RVDB. So the story back like decades ago, when we were using the public domain database to detect the rhabdovirus in SF9 cell, and we figured out the problem because by that time we took the public domain database such as NCBI RefC. The problem for RefC is that yes, it's well annotated, but since the sequence quality is really good, and all of the sequencing RefC that is created by NCBI staff. So you can imagine that in millions of the sequence which is already available in GenBank, the NCBI staff cannot quality check all of these sequences one by one. So they have some sort of the criteria to decide if they can promote this sequence to the higher level, which is RefC. So in that way, the RefC, yes, the genome quality is really good, and you can have the available annotation, regarding the function of the genome. But the problem is that the diversity is just not high enough. You may have only like a 1 or 2 representative sequence for each species. And then we say it's okay, then we go to NRNT because theoretically NRNT has all of the sequences that's deposited to gene bank. So in that case, we should have very high diversity, so we have the chance to detect more unknown viruses layer. But we figure out another problem. Because gene bank, yes, the sequence depositor, they deposit sequence layer. And I won't say all, but few depositors, they might not be careful enough. So when they prepare the sequence, they write a paper and they deposit the sequence, lots of things could happen. For example, they forgot to trim out the adapter sequence, or they forgot to trim out their plasmid, or they somehow do the de novo assembly, so they accidentally assemble the virus genome with the host. So if you directly take the gene bank, you will see tons of false positive hits because of this. Especially when you are dealing with, for example, you are taking the samples from the virus C with a very high host background layer. In that case, your high host background will be aligned to this gene bank if that sequence is not annotated correctly. And we have search tools to use NCBI protein database. Yes, that is another choice. And actually, this choice will give you the higher chance to detect any unknown viruses because the protein database is essentially a functional database, right? But the problem is that the protein database will only show you the coding sequences. So for example, if you have the virus sequence, you have the read that's sitting in the untranslated regions, you will not able to get that. So what to do? So the work is initiated back in 2013. Our lab has the in-house effort with the AVD TIG balance, we call it TIG. And right now it's TWG and maybe become TEG, I don't know. And also we work with the Ronnie from NCBI, try to come up some idea, how are we going to have a high quality sequence in one database while the diversity is high enough to give us the higher chance to detect all of the unknown viruses. And we conclude that we should have all viral-related and viral-like sequence layer. And we also include the endogenous virus and the retro elements back in that time. Meanwhile, we want to have the database with the as less as possible host carryover there. For example, just like I tell you, somebody may misannotate it. We want to do some manipulation or by some bioinformatics to get rid of these sequences. And the initial work of RVDB was published by Norman in 2018 in NSphere. And hopefully the newest, we have the update, including all of this remote work, and hopefully it will publish sometime. Everybody say next year, so I will say next year. So how do we make RVDB? So actually it's quite simple. We take the sequence from NCBI Gene Bank, NCBI Viral RefC, and also NCBI TPA. And Norman developed the strategy to identify the non-viral sequence based on the FASTA description. So we have about a bucket of keyword. Right now it's like say 700 keywords. That is pretty much based on our knowledge accumulated during these 10 more years. We do the manual curation for each update so we can have sense of what keyword might give us the hint that this sequence might indicate to non-virus. So we do have this keyword, and we have this keyword list, and nomenclature is the semantic refined filter or SENR. So basically, there's a Python script to have this native keyword list, and you have Python script to iterate to scan all of the sequence. But for GenBank here, we include all of these divisions. That the idea to keep not only the viral sequence, but also the lots of another sequence from another division is that we, like I said, we want to keep the retrovirus and also the retrotransposable elements. So in many cases, these retrovirus won't be classified as viral because they will be classified in their host. But here we intentionally not including the phage because we already know that phage, lots of the sequencing vector or adapter, they do have the phage sequence there. So if we include this phage, we will again give you some false positive hit. And then we have this candidate sequence. Once we get this candidate sequence, Arifa and I will sit down together to computer screen, and we use our human eye to check one by one. And every update, we will have like 100,000 newly added sequences. So you can imagine that's lots of work. So that's why in my last slide, I will tell you how machine learning tool can sort of help us to reduce this work. And RVDB right now, if you go to the RVDB site, which I will introduce, and you will find four format there. One, the first format is the unclustered RVDB, which means we collect all of the viral sequence there, okay? So it is suitable for if you have a very high power computer, you can use this file. Right now, I will introduce later, it's really big. So it will suitable for virus detection by such as +N or HNMR. And for some of you, you might not have such a powerful computer layer, then you probably won't able to run the URVDB layer. In that case, we take URVDB and then we shrink the database size by doing the collapsing. And how this collapsing does is that we scan all of these sequence and we pick the longest one to compare with other sequence. If the other sequence show at least 98% similarity, and those subset of sequence will be collapsed by its representative. So it's dramatically reduce the database size. So we have CRVDB, so that's suitable for virus detection by T+X because you know T+X need to go through all of the 6 open reading frames. So if you use the T+X to run URVDB, it will run forever. So you don't do that. You use the CRVDB to run T+X. And for some more advanced users, they might need to use the RVDB to create some database. We provide the SQLite file. And Mark, their effort is to provide the Proti version of RVDB. And it is suitable for HNN scan or HMN search or HMN scan. So RVDB current version is 29.0. So here's the QR code if you are interested. And the newest version is released in this summer. It's based on the GenBank release this May and also reference release this RefC release this May. And for the uncluster, just I tell you, it's really big. We have like a 10 million sequence there. And the cluster one, by collapsing the sequence, share the 98% similarity, we only have 1.1 million of the sequences. But you can see during the past 4 years, because of the COVID pandemic, researchers are crazily submit their SARS-CoV-2 sequences. So you can see more than half of sequence in URVDB are essentially SARS-CoV-2. And the RVDB is available on University of Delaware site, and also, like I say, Mark, they do the protein database, which is available on Institute of Pasteur. So how about refinement? Like I said, because we directly take sequence from NCBI, so any kinds of the error they make, we carry over. So it's not our fault, their fault. So it's included poor quality of sequence. For example, we do see lots of poly-N in SARS-CoV-2. And also, like I said, we have the sequencing vector carryover. For example, you will see that sequence is the depositor said that it's virus sequence, but we do see they are now trimming out the vector, adapter, linker, primer, something like that. And also, there are some cases this sequence is not virus sequence at all, but somehow they annotate it as a virus and vice versa. And especially for the endogenous retrovirus, there's a case that when people are submitting their sequences, they didn't really carefully check. So you will have the middle of the sequence, which is belong to the endogenous retrovirus, but they do have the host franking in 3 and 5 prime. So we have some pipeline strategy to develop in past few years to address all of these problems. One thing is to update our bucket list of the native keyword list. For example, here we designed, all of this list was discussed in MVDT WG subgroup C. So we decided to discard if the FASTA header indicates that it's an untranslated region or UTRs, because you will, if you have any chance to see the gene bank, and you will see lots of these sequence without the actual viral coding region there. The idea is that people submitting this UTR, I think they are for the phylogenetic purpose. But we decided not to harvest this sequence because the actual viral sequence are available. And that is in the full length. And we also decided not to keep the satellite sequence or satellite viruses because satellite sequence or satellite virus, they need the helper virus to replicate. And again, we don't want to keep phage viroid because, as I say, it has the similarity with the sequencing adapters. But we do keep viral phages. And also, especially in environmental sample division, you will see lots of FASTA description saying there's, for example, unverified. And we have an intensive discussion about it, and we decide to not having low sequence in RVDB because there's just no way if you have the heat for these no-virus defined sequences, we don't know how to follow up. So that's why we decided not to keep this. And also we have this stiff virus, it's not truly virus at all. And also by menu curation, we identify some non-viral sequences. And some people, they didn't really follow the consensus. For example, we are supposed that TY or PO are used to name ritual transposon, but somebody actually use these two terms to name their clone. So we had to going through all of this menu curation to identify if this TY or PO really designate to the ritual element or people just use it to name their clone. And also we get rid of the host of their IAP. So I think this part will be most interest to you because we have lots of discussion about how to increase the sensitivity, not necessarily sensitivity, but how to increase the specificities. And we have lots of the discussion here, and Chen and Mehdi, they do a wonderful work that they took the entire RVDB, to run the blast against their in-house knowledge-based non-viral data set to identify for all of the sequence on RBDB if there is any fragments in this sequence that share the homology with the non-viral sequence, they will give you this annotation sheet, okay? So here is the example showing here. So we have the query sequence and we run the blast. We figure out, oh my God, we have HIV. What happened? And because this HIV somehow share the similar homology with these two segments here, which is come up to mitochondria. I think, Christopher, you like this. So we provide this annotation sheet. If you go to the RBDB site, there's a download link. So you can, when you run the NGS and you use the RBDB to analyze, if you see something pretty weird, you see SARS-CoV-2, you see HIV, you see whatever virus is not supposed to be there, please just go to the annotation sheet, because this annotation sheet will tell you for this sequence in RBDB from where to where we detect this is not only virus, but also some non-viral sequence, and we give you the category about what are those non-viral sequence belong to. And another refinement, the first one might not be relevant to you, but we work with JSO in their University of Delaware, because originally the RBDB production script is written by Python 2. So, and very few cases, some people are interested in making their own RBDB, and we do provide the RBDB production Python 2 script on GitHub. And because the Python 2 is already obsolete, so we have the help from JSO to help us to transit the Python 2 script to Python 3. And also the original RBDB production script is really long. I have to run different scripts one by one. So I just sort of consolidate from 85 Python script down to just 2. So it saved me lots of time. And also the second improvement is the taxonomy-based phage cleanup pipeline. Because like I tell you, we use the native keyword to address the phage sequences. But right now, people are naming the phage in different ways. For example, we used to name the phage as mycobacterium phage, but right now people call it mycobacterium virus. So in that case, because we don't have the phage as a keyword, so we will not able to catch. So instead, no, not instead. In addition to having the native keyword ACNR layer, we also implement the taxonomy base, which means I take the phage taxonomy and scan through all of the database. The third one is poly-N detection and filtration pipeline. That is only for the SARS-CoV-2. So you cannot believe there are 100%, no. You can search NCBI SARS-CoV-2 and you will find hundreds of them have the full SARS-CoV-2 sequence, 35 base pair long. They are all N's. So I don't know what that happened when we asked NCBI and say, oh no, it's the pastor's choice to retrieve their sequence, but we still need to keep there. So it's okay. We have the pipeline to get rid of this poly-N sequence. And also, especially for past 4 years, we have the trouble with collapsing the RVDB because the database is just too big. So we come up with some strategies, which I show here. So this benchmark show you how the redundancy of SARS-CoV-2 could impact your analysis, okay? But if your institute is using the K-means strategy such as Kraken Unique, actually the impact is not that significant. So here I have the entire RVDB, and also I have the class RVDB, and I have the entire unclustered RBDB, but I intentionally only keep one SARS-CoV-2 sequence there, which is the Wuhan strain. So for the camera-based strategy, you don't really see much of the performance difference here. But if you like us, we like BLAST-M because it gives us the superior sensitivities. That will cost a lot of time to run the entire RVDB because you can see the time difference is around threefold compared with the entire RVDB and entire RVDB, but just with one SARS-CoV-2. And also it costs lots of CPU usage. So what do we do? So here is our new collecting strategy. We have the candidate sequences, and then we pass through the SARS-CoV-2 sequence with poly-N filters. So like I say, if the SARS-CoV genome has more or equal to 1% of poly-N, we just don't want it. And if we keep the sequence below 1% of poly-Ns, and then we take all of this sequence, align against the only one reference SARS-CoV-2 sequence, which is the Wuhan strain. And if this alignment gives us more or equal to 98% identity, all of these sequences will be represented by this Wuhan strain. But if the sequence is less than 98% identity, all of these sequences will be collapsed and generate the CRVDB, but only with SARS-CoV-2. And this collapsed RVDB with SARS-CoV-2 will be combined with whatever the non-SARS-CoV-2 sequence is go through the collapsing step and give you CRVDB. So here show you the performance of the SARS-CoV-2 collapsing. So initially, without any collapsing, we have 5.5 million of the SARS-CoV-2 sequences. And we pass through the poly-N filter here. You immediately get rid of 1.7 million of SARS-CoV-2 sequence, which has very low, I shouldn't say very low, it depends on your criteria, but with more than 1% of poly-Ns. But if you think about it, if you have certified base pair, certified KB, and 1% is a lot. So it further reduced it. And we further collapsed by the Wuhan strain, and it further reduced the SARS-CoV-2 sequence to just 1 million. And this sequence will be further collapsed if not sharing the high homology with the Wuhan strain. And finally, it will only give you 1,000 of SARS-CoV-2 sequences. So totally from here to here, the collapsing ratio is about 5.2K. And again, SARS-CoV-2 sequence is really big, so it occupies lots of size. So we can reduce the size from 159 gigabyte to just 3.1 megabyte. So it's like a 51K times. But you may imagine, okay, we collapse the SARS-CoV-2, but how about the diversity? Yes, we do have the sacrifice with diversity because I determine the Pango lineage before and after collapsing. It's like 64 less. And also the unique Scorpio code is 3.1 times less. But compared with the size you save, that definitely gives you some balance between you can detect something under a reasonable time. Yes, the collapsing of SARS-CoV-2 sequence, we sacrifice some diversity, but after the collapsing, we still keep majority of the variant of concerns, VOC. So you can see we have still keep alpha, delta, gamma, mu, and Omicron strain in CRVDB, yes. So I don't have the update for the unique user of RVDB for this year. But last year, we have 5,000 users, and majority from US, China, and other countries such as Europe. So what are you waiting for? So the current and future work, we continuously have the in-house effort for RBDB updating and refinement. We do promise to update RBDB quarterly, so we will have 4 updates per year. But this year, and I would say last year, is quite special because of pandemic. So we are struggle. ABD TWG subgroup C struggle a lot to figure out the way how to collapse this SARS-CoV-2 sequence. And this year we have another challenge because of NCBI. They start to providing some essential file that is necessary for us to make RBDB. So, but I'm happy that things so out. So hopefully RBDB version 30 will be available before this year. And also we are going to keep annotate the non-viral and viral, but with host content. The way to do that, like I said, Chen has the in-house non-viral data set, and he is going to expand this data set to give us a higher chance to detect the non-viral sequence inside the viral genome. And also, we are planning to have the annotation of the whole sequence in viruses with large genomes, such as herpes virus and Pandora virus. And I think this one will be challenged because right now the URBDB or CRBDB, they have both exogenous virus and endogenous virus all together. And we try to figure out a strategy to separate these 2 sequences in different bins. So in the future, you will be able to choose what version of the database you want. And also we are going to, we have the RBDB user survey back in 2020, and we get lots of feedback regarding how to improve the RBDB use experiences, something like that. And we are planning to have the same, probably based on the feedback and also the refinement, improvement we have done for these few years. We are going to have another survey. And last one is the prioritization. It might not avail you a lot, but it definitely helps us to make the RBDB production turnover more effectively. And we are going to, right now the RBDB production script is single thread, which means it cannot take the advantage for the current modern CPUs, which has multi-cores. So we are going to rewrite, or I shouldn't say rewrite, but modify our current Python script to implement this modern CPU. And we already finished one script, and I can tell you the performance is amazing. So stay tuned. Machine learning. Okay, just like I said, for each update, we have to sit down in front of the computer, look at this Excel sheet. That is 100,000 rows. And then we figure out, okay, right now everybody is doing machine learning or AI. How can that help us? And we do keep all of the sequence FASTA header. Arifa and I think based on our biology or virology knowledge, we think that is non-viral. So we use this data set, we label it, we do the supervised learning to feed the training data set, and also we evaluate this model, then we justify the performance. And I can tell you, we tried like 7 large language models, and all of these 7 perform great, but one model stand out, which is BERT. So we use this BERT model to further test if it really works. And like the example I tell you, somebody used TY to name their clone. So you can see this yellow highlighted. They are not truly TY retro transposable elements. And the BERT model is smart enough to tell me, okay, this is negative. And rest of these TY, they indicate to the retro transposon, they give it as positive. So I think there's a potential there. So we are investigating other models which has higher parameters. So we are acquiring the GPU with more range in order to accommodate all of these modern LLMs. So I would like to thank my advisor, Riva, and my colleague, Chen, and also Mike helped us to parallelize RVDB. And also the University of Delaware team, Sean, Jethro, Mehdi help us to host the RVDB database and also do the refinement. And also, of course, the ABDT WG subgroup C. And this funding is from the FDA, Purdue, and also the MIMBO. Thank you.
So firstly, I'd like to say thanks to the committee for giving me the opportunity to present today. And secondly, apologies for not being there in person. So this is next generation sequencing, our approach to validation, regulatory approval and implementation of next generation sequencing as an alternative to the in vivo method for FluMist and FluEnz. So I know this will be a lot of repeat around what people have said today, but next generation sequencing, why? So really we can use it to characterize any adventitious agent that may be present and optionally confirm the genome identity of a virus-based test article. Next-gen sequencing, as we all are aware, is seen widely as an alternative to in vivo and potentially in vitro testing. The in vivo test is outdated, not sensitive, not robust. And, you know, European Pharmacopoeia are going to introduce a chapter for NGS, as well as obviously the ICH, which has already become effective. Aligns with the 3Rs and sustainability initiatives, and allows us to move to a much more technologically advanced analysis for adventitious agents testing. In our case, it dramatically reduced our turnaround time. We also had a very high percentage of invalids when we were performing the traditional in vivo testing, and that was just due to our own flu sample matrix. When we were starting to look at this, we quickly realised that we didn't have the lab capacity to develop and validate NGS internally, so we entered into a collaboration with Merck to validate the NGS method for FluMist and FluEnz. So I worked very closely with Brad and his team to get this done. So I'm only going to quickly go over these slides because I think everyone, you know, knows all the different regulatory guidance from the European Pharmacopoeia, US FDA, and also the ICH-Q5A R2. So when is it appropriate to use NGS technology? So really, if you've got an infectious product or intermediate where a neutralizing antibody is unavailable for culture-based methods, cell line screening in lieu of animal studies and in vivo testing, broad virus screening to complement or round out the safety profile of the test agent, product release testing as an alternative method to confirm safety profile, instances where traditional assays would not be expected to identify a potential contaminant of interest or no traditional assay exists. Replacement for in vivo and in vitro methods where appropriate, consistent with our 3R principles, and provides faster alternative results to traditional testing. So really, before we went into this, we looked at what our considerations would be. So we wanted to be able to, you know, look to see if we had any adventitious agents to replace the in vivo testing. We did know that, you know, if we did detect some nucleic acids, but may not be infectious particles. However, the information gained through the sequencing would be able to guide our investigation path to look at virus identification, whole or partial genome, and known replicas of intermediates. More considerations for us were also the method quality. So, you know, we wanted to establish criteria for method suitability, including the sample preparation, the assay limits, and the process controls, how the data was processed and stored, a defined bioinformatics approach, and data interpretation in the context of the sample tested. So the next few slides I'm going to go through will break it down into our validation and sample matrix qualification, the regulatory submission approach, health authority questions that we received, health authority approvals, and what our implementation strategy was. Again, not going to spend too much time on this because Brad previously presented this as well. Obviously, because we were working with Brad and his team, they do have an NGS assay which is validated in the modular fashion. So extraction, library prep, sequencing, and bioinformatics. The limit of detection we determined through product-specific qualifications. We did use the panel of 5 viruses, the WHO reference material. Our product-specific qualification was completed in 2022, and it was just the 5 viruses that were available at the time. So a little bit about the sample matrix. Our overall goal was to replace the in vivo animal model. The actual matrix is bulk viral harvest of a quadrivalent influenza vaccine. So we split it into 4 separate strains and lots. So a H1N1 strain, H3N2 strain, a B Yamagata strain, and a B Victoria strain. Our actual vaccine is manufactured in SPFX as per for European Pharmacopoeia 522 and USDA Memorandum 865. So just a little bit about the actual QC release testing. So we do a lot of QC release testing, as you can imagine, on a drug product. We test on all different stages throughout the manufacturing process. Our testing is designed to assess the safety, purity, identity, and efficacy of the drug substance and the drug product, with our main kind of points of references being 2616 from the European Pharmacopoeia and the US Guidance for Industry 2010. We performed in vivo testing on every single batch of drug substance that was manufactured, was subject to very high invalid rates due to inherent variability within a biological model, egg hardiness, or all that good stuff when you're working with in vivo testing. So we were looking for a suitable alternative, which was validated, which could deliver and scale for patients with quality, speed, and efficiency, supply to market with a repeatable, robust assay, accelerate our digital and technical innovation, and also in line with removal of animal testing in line with AstraZeneca 3R's program, which is why we decided to proceed with next-gen sequencing. This data has already previously been shared at the previous IABS conference 2 years ago. At the time, this was kind of hot off the presses because we had just literally completed the product-specific qualification. But a very brief kind of overview is that we started at 1E6 and went down to 1E2 virus spikes in all 4 different strains, using the 5 viruses from the WHO, but also another virus of interest that we'd added in as well. So this is a very high level summary of the results. So you can see, results for the 1E6, 1E4, 1E3, and 1E2. All of our assays and controls passed validity criteria as expected, and we had no hits, if you will, in the unspiked samples. We then combined the LOD, and we had detectability for 4 viruses at 1,000 genomes copies per mil, and for 2 viruses at 10,000 genome copies per mil. So the conclusions from our spiking study was that NGS was able to detect representative virus spikes at suitable limit of detection, and that was in line with previous matrices published through worldwide studies. Results were in line with expectations regarding limit of detection between 1E3 and 1E4. The higher limit of detection in the reovirus and the porcine circovirus wasn't unexpected to us, given the nature of the viruses. And our read numbers were greater than 100 million reads per sample, were ideal for hit and detection limit based on hits per million assessment. The minimum read count set at 130 million for this matrix. So this is really where I want to concentrate the rest of the presentation. So this was our regulatory submission approach. And just before I go through the slides, just another little bit I think I'd like to add in is that at the time, we did not, as a company, have any technical discussions with the FDA or EMA or any of the other markets that we were submitting into. And in hindsight, I actually wish we would have done that now. We put together what we thought would have been the best submission approach. But when we actually look at the questions that we got back, if we'd have spoken up front, say with a REFIS team or people in the EMA or MHRA, we could have actually provided a lot more data up front than what we actually did. So really, we put together a summary validation package, which Merck provided for their overall modular validation approach. Also note as well that Merck do have a file with FDA with all of their validation documents in which FDA were able to review and see some of the proprietary information that couldn't be shared with us. We then put together a final summary report between Merck and AstraZeneca detailing the overall product-specific qualification approach and all of the data. We also had a contamination control assessment of manufacturing and the QC labs, really to show how adventitious agents couldn't be introduced during manufacturing or the testing. We then had a full risk assessment for the introduction of next-gen sequencing as an alternative to in vivo, and this included the contamination control of adventitious agents. We put together a comparison of next-gen versus in vivo. We had a section around risks of switching to next-gen sequencing, another assessment on the safety profile of the actual product, and a discussion of the other safety tests that we do perform. We also put together an investigation approach for if we were to see a positive hit in routine testing. And overall, the summary gave us was that NGS offered us a more robust, sensitive method, along with our other registered safety assays for the detection of adventitious agents. So it wasn't really a case of, well, we've got next gen, we're now going to stop all of our other safety testing. We are still continuing with that alongside next-gen sequencing. So a few examples of questions that we got from various health authorities. So the first one being that the applicant should provide a detailed description and justification of the primary algorithm and secondary analysis, and the criteria leading to the definition of a hit and a true positive viral hit. So myself and I worked with Brad and his team on how to respond, how simple things of how the dilutions of the virus stocks were actually prepared, questions around the matrix, the material that we'd used in the spiking studies, and if the actual pooled harvest fluid was representative of commercial lots. There was a comment around the LOD of Rio and porcine circovirus being a log higher, and a recommendation really to continue to improve the LOD of those families. And then there was questions around the rationale to determine the following viral signals as not being considered significant. So viruses such as marine and fungi-specific viruses, and plant and host other than Gallus. Then there was questions around the detection of other influenza strains which were detected in the Lave strains during the blast results of the PHF. And that's literally, it's really around the backbone of our product. So the internal genes being very similar across different flu strains. And then finally, please submit plans for updating the validation studies. As you know, it has been mentioned that the workflow can still be optimized and the database and bioinformatics software are evolving. So kind of that and the LOD of the Rio and the PCV were kind of the theme of, you know, yes, it's validated now, but what are we going to do to continually improve the actual assay that we have validated? So questions we received were from EMA, Swissmedic, and the FDA. All questions were responded to within the boundaries of our active submissions under review. So far, markets that have approved are EMA, MHRA, Swissmedic, Ministry of Health in Israel, Health Canada, and the FDA. And we currently have next-gen sequencing under review in Mexico, Taiwan, South Korea, and Australia. We are also currently in discussions with Japan to understand if we can submit next-gen sequencing to Japan to see if they will actually review and approve the next-gen sequencing method. At the moment, we're still being told that in vivo is still the chosen method. But as I say, we are in discussions with Japan around how to potentially move this forward. And what do we need to provide to satisfy Japan's regulators around next-gen sequencing. So our implementation strategy, next-gen is now the preferred option for adventitious agent detection for flu mist influenza from an in vivo perspective. We actually implemented next-gen as the alternative method for in vivo in April 2023. So since April 2023, we have had 0% invalid assays for next-gen sequencing. And when I put that in relation to in vivo, where at times we could have an invalid rate as high as 50%. I think a real positive, I think, for NGS as well is that we've had 0 positive hits which needed to be investigated from April 2023 to the present day. A big positive for us, our turnaround time for next-gen sequencing is 32 days. Our in vivo turnaround time was 60 days. So that is a massive 28-day turnaround time saving, which when you're working with a seasonal flu vaccine, literally every day counts on being able to get your product out in a safe and efficient manner. One point I would like to really go over as well is that next-gen sequencing is more costly than in vivo. However, we have balanced that with the fact that we have a 28-day reduced turnaround time. As I say, 0% invalid assays, which have needed investigations. And up to this point as well, fewer investigations for unconfirmed outer specifications. You can imagine if you've manufactured 50 batches, 25 of those batches have got unconfirmed OOSs for in vivo testing, the amount of man hours and time that it takes to investigate those and potentially go to a retest or a repeat and getting further data. You can imagine how many hours that actually takes, not just from QC, from QA, all of the supporting functions as well, which are involved in those investigations. But just a point to note that we are still performing in vivo testing for some specific lots for markets not yet approved. So in particular, we're still performing in vivo for Japan. So I'd just like to thank Brad, the regulatory team, and the entire NGS team at Merck as well. And thank you for your time.
We have about 30 minutes. panel discussion and questions. Yes.
Thank you for all the speakers, great talks, and very interesting results. So I have three questions to three speakers. Maybe the third question will go back to the first speaker. First question is for Bradley. So in one of your slides, you mentioned that for one matrix, if I remember correctly, is the insect cell harvest that tend to be a difficult matrix. It's low sensitivity, even you use benzonates. Is that insect viral harvest matrix?
Yeah, so it was
Okay.
an insect viral.
So you think there
harvest.
is the benzonates works, but it just the matrix has something that the benzonates cannot reduce.
So it was the virus. So that particular matrix contained high levels of baculovirus in
Okay.
there. And so the benzonase doesn't take care of the baculovirus. And so that's what we believe is causing the higher limit of detection.
Thank you. And the second question is to Valeria. So from the different labs, the sequencing depth vary from 8K to 40 million. Is the input library yield positively correlated with the output?
Thank you for the question. There are several, several steps and several things to take in consideration, because once, of course, will be the input of the library. Then we observe also variability among the different batch-to-batch flow cell, and so the different quality of the number of pore that are available, and this could impact also the number of the sequencing that are producing during your run. Another point to consider is the chemistry, because we start and at some point some labs perform experiment with a different chemistry, and different version of the software and the base calling. And so this is another issue and another point to consider if you want to compare the different data that were obtained. So we have to evaluate once the complete package of data set will be available. Maybe we will able to try to align and find some critical points that could be improved and could be taken as consideration when you apply this technology.
Okay, thank you. Yeah, I think that would be a very interesting data set. I have like 11, 12 labs. Maybe you can apply some
12.
machine learning model to identify the feature that impact the quality of the rays and the output. Essentially, you have more output, you're going to detect the sensitivity going to be better. That's,
Right.
I think, I expect.
Yes.
Okay, thank you. And the third one is to Alice. So in your talk, you mentioned that the PCV1 and Rio has 1 log higher sensitivity compared to other spiking viruses among the WHO viruses. So I'm wondering, like, PCV1 is the smallest genome among the 5. Do you see that in other matrices, or do you only see that in this matrix? You have a better sensitivity actually for PCV1.
Yeah, so I think this should probably go back to Brad. The reason, sorry, Brad, you love me really.
No problem.
The reason being, I've obviously only validated for my sample matrix, whereas Brad has validated other sample matrices and would be able to comment on if he sees that with other sample matrices. Sorry, Brad.
Yeah, no problem. So we do see a lower, a worse, what is it?
Oh, sorry.
Higher limit
It's
of
higher IOD.
Yeah.
Okay,
so-
that makes
yeah.
sense.
So
I
we
saw
do
there's
see a higher
higher
LOD generally
sensitivity.
with
Okay.
PCV and Rio. So
Yeah.
PCV is your really short single-stranded
Right, right.
DNA, 1.8 kb, and then Rio being your double-stranded segmented virus. We tend to see a higher LOD with those 2 viruses when you compare them to the other viruses.
Okay, yeah, that makes sense. But Rio, maybe you want to open up the double-stranded RNA more. to improve the sensitivity. Thank you.
Hi, Charlie Swafford, MIT. Great talks. Valeria, you're very close to the edge of the stage. Trust me, it's a doozy. My question is for Brad. And really anyone can comment on this, and it really goes to the comparison of the sensitivity between PCR and NGS. And in the data that you showed, there was a lot of variability due to the different matrices that you were looking at. And I guess I was curious if the PCR, if you did, I think you did PCR on that as well. Correct me if I'm wrong.
And we didn't do PCR.
Oh, okay. Well, maybe you can just, I guess what I'm curious about, and maybe this is a more general question, does that sensitivity or does PCR correlate with those changes in sensitivity due to those matrix effects as well? So, I mean, if we're going to say that PCR and NGS are comparable to each other, I would expect that if one matrix is particularly difficult from a sensitivity standpoint for NGS, does that hold true for PCR and so forth?
So I'll go back to my old molecular days where since it's exponential amplification in PCR, you're generally able to amplify that above the background, right? So if you have a higher background with NGS and a lower, higher limit of detection, generally we didn't see that with our PCR assays. Going back a couple of years, you can add up to a microgram of DNA in PCR and you'll still see that, X number of copies, however many copies that is. Whereas with NGS, that background would suck up all your reads because you're not amplifying. But I don't know if anyone else has anything to.
Yeah, hi. So maybe I can comment on that with regards to the study that Allison presented. So, she did mention that in the pre-study, the ddPCR was done just to identify the range of samples to test. So there, we did find that, and this is done on the same sample that was run in the NGS study, so that there the sensitivity was very close between the ddPCR and the NGS, but in that I think it may have been 1 log better compared to the best of the NGS data that you saw there. But it was close. But again, and that was in a pretty complex matrix, the unprocessed bulk. But clearly, I think we're all aware that the sensitivity of PCR also depends on the background, depends on how much test material you're using. If you go over 0.5 microgram, you're going to get inhibition, less sensitivity. And I guess the more general question about, I know we're challenging NGS for sensitivity, limit of detection, demonstrating, it to a higher bar, I think, than we sort of put PCR at, in a way, because when we get PCR results, there's a fairly wide range based upon your assay, and I don't think that there's established expected sensitivity for PCR assays in general, although the assays are validated. So just wanted to comment on that.
Yeah, I have a question for Mark and Peju. I'll start with Mark. I don't do transcriptomics myself, but I was wondering how specific are the messenger RNAs when you do transcriptomics? Can you attribute them to a species level, or how does that work?
It depends on the sequence. In fact, the pipeline we are using returns the lowest common ancestor, meaning that if the sequence is shared between different species, it will return the genus. If the sequence is shared between different strains, it will return the species, etc., etc.
Okay, so you expect to find, if you would find novel viruses, that it would also share that concept. So, okay.
Exactly.
Okay, thank you. And Peju, the example that you had with the HIV being associated to mitochondria, probably, it looked like to me immediately as a bed file almost. Could you rewrite that to mask these sections so that you would not get hits on them?
Yes, our purpose to provide this annotation sheet, if you have any chance to go to the University of Delaware site, we provide the annotation list just like the example I just showed. And in this sheet, you will find the accession number in RVDB along with any non-viral region inside, and we give you the coordinate. And we also have this discussed internally. Should we go ahead to mask this region in RVDB and release to the users? But it sort of become our responsibility. You understand? So, but you have to keep in mind that I'm not saying whatever in the annotation sheet, you have this non-viral, we shouldn't call it non-viral region. We should call it is maybe viral or non-viral, but simply this region share the similarity. We are not sure if this region is truly non-viral. It's simply they have the similarity layer. So yes, I think it depends on your risk and also how you implement the bioinformatics. If you think that will make your life easier or that will be more practical for you, you can just use this annotation sheet and mask your own database. So as the database keeper, especially this database is kept inside FDA, so sort of like the liability issue that because we cannot validate if this sequence is truly virus or non-virus. I think we provide the purpose here is just to warn you if you see something positive, but you also, based on your own risk management inside your facility, you think this kind of positive piece shouldn't be there, then we provide that annotation list for you to do the follow-up.
So you could basically use it as a screening afterwards too.
Yes, you can do that. So do you want to add?
So Pero, I know you're being cautious, but I don't think that you need to feel that there's lack of confidence that is non-viral, because there is secondary screening that Trent does. And he takes that region and then he hits it back against NT to verify that it's not picking up any viral sequences. So I think, I realize you're being cautious. And of course, there are many viral sequences that have relatedness in small regions to non-viral sequences. So as a caution, I think, we can put that disclaimer. But I think there is confidence that everything that has been done, bioinformatically does indicate it's non-viral. Plus the data sets that Trent has developed for non-viral sequences, he's tested those data sets against RVDB and well, against viral sequences also, and they don't contain any homology or any hits to viral sequences. That's the reason we haven't really made a cellular non-viral database like a mammalian cell DNA or something, because there we know that there may be viral sequences no matter what genome you pick. So if you have to first screen that genome, with a brush, and then make sure that there's no viral sequences in that non-viral data set, especially if it's a DNA, cell DNA data set, because otherwise, if you use it, then you're also doing non-viral sequences. But the masking also, there is a concern, because I think once you mask it, and I know people do that, they many times mention that, but to me, I guess once you mask something, you may have to demonstrate that didn't mask any viral sequences. And so I think that's the reason we don't want to take that responsibility also.
Okay, thank you very much. I have a question and a few comments for Alison. Laurent Mallet, DQM. Thank you very much for this very interesting presentation. I think it was very, very interesting. I have to say that it's always difficult to conduct a head-to-head comparison and a comparison in general. So I think it's a challenge. I have maybe a few comments regarding the fact that you will always find viruses which are maybe better detected by cell culture or in vivo. But of course, in the comparison exercise, you need also to take into account the breadth of detection of the NGS, and this is what you said as well. So I think we have to have a balanced interpretation of the results, because of course you have to select viruses where you can compare something, so which are detected by the in vitro or by the in vivo to make a comparison. I have also a question. Can you confirm on the reovirus what was obtained? It was positive by CPE, negative by hemadsorption, positive by hemagglutination?
Sorry, correct. Yes, that's right. But only using the rhesus monkey red blood cells. We used other species. But that was the only red blood cells that actually came up positive. But the Rio was all CPE, no problem. And then the hemabsorption assay. Sorry, my apologies, hemagglutination.
Yeah, okay, thank you. And will you consider that your viruses were cell culture adapted already, or they could consider as more wild strains of these viruses? It's just a question. It's not at all to challenge it. It's just that, of course, there is always this question behind it.
So it's the reovirus type A, strain 1, which is used. Sorry, Anif is jumping in here because it's obviously from the materials that are used for the reference database.
So the strain is human type 1, Rio Lang strain, and it was from ATCC. So of course is cell culture adapted. With regards to the Rio, and again, it's in the literature, but there are many Rios that are CPE negative, but they're still HA positive. And the one we used happened to be CPE positive, but we didn't really, select it for that purpose. We selected the virus because of the genome structure.
Okay, thank you very much.
Yeah, hello, Noémie Deneyer from GSK. I have a question for you, Brad, regarding it's linked to the question you had paid you this morning about maybe the platform method validation for cell-based sample. Because so broadly, so you presented that indeed you have like your method validated, and so you're applying for different cell lines. So my question is, how are you assessing this cell type background? Because we know, for example, that SF9 can come with a rhabdovirus, that, for example, there is some cells cultivated with serum, other with non-serum, etc. And so how are you adapting then your analysis and your report based on that?
Right. So no, that's a good point. And anytime you run NGS, you detect sequences, right? So in any cell line, you're going to see sequences. So it's important to know enough as much information as possible about what you're sequencing. And so that way you can put the sequences within the context of what you're analyzing. And so for SF9, we do see the rhabdovirus, we do see the characteristic sequences that you would expect to see within SF9 and that are supported by the literature. And when we did our limit of detection study, what we were looking for is the 3 virus spikes to make sure that that cell line, the unspiked sample, we did not see the 3 virus spikes. And for the spike sample, we do see those because that speaks to the specificity of the method to identify those particular ones.
Okay, so it's really based on the literature then. You are doing like an extensive analysis of the literature for the cell line that you are analyzing, and then you are adapting based on that.
Yeah, I mean, we don't do host cell filtering, so we do see endogenous retrovirus sequences, and we call those out. So we call out all of the sequences that we do see, and we'll place them within context of the cell line that we're testing. Now, this particular assay isn't designed to detect endogenous retrovirus. We identify those sequences, but using more of an assay like a PERT assay or a TM assay or something like that to complement the NGS is really what would put those into better context. But yeah, we do see a lot of background.
Just had a quick question for Brad. Just clarification, when you were discussing the matrix effects, we see an LOD difference, I think, between the first and last. It's like 1,000-fold maybe. From what I'm hearing is that is primarily due to the virus production itself in the background. Is it all entirely due to the virus production, or is there a contribution from something else in there?
So no, that's a good question, and it depends. There are sometimes a lot of host cell sequences as well, but you can remove those with benzenose treatments or filtration or other treatments. And so that the data that I showed, we'd already optimized whether or not we use benzonase. So those sequences were actually coming from the virus. And when we investigated and we mapped those sequences to see where exactly they were coming from, in the baculovirus example, I'll give that because that was the worst case scenario, we found that the majority of those sequences were actually mapping to the baculovirus.
I have a technical question for page 2. So you mentioned that there were some difficulties with including phages because of their resemblance to some vectors. Some animal viruses resemble vectors as well. So how does RVDB manage those?
So you mean the amino phage, or sorry, can you?
Animal viruses
Animal
that you
vi-
do include in RVDB, some of those resemble vectors as well. And so how do you manage those?
Yes, so that's we have the annotation sheet that if you see this non-viral data set keep in house, we also have this vector sequence there. So in your case, if by any chance the sequence in RVDB, the virus from animal, that sequence has any vector carryover, theoretically by the annotation pipeline, those vector fragment will be annotated.
Okay, and so is the reason for not including phages because that was a manual process before? Like, is there hope that phages could be included in the future?
That will become a question for subgroup C. Yes, we can.
Sure.
We
bring
can discuss it because I guess yes, I agree because the phage itself may give us the broader view about the contamination maybe from bacteria, but we really need to have a broader discussion to judge the pros and cons to include the phage sequence there. But yeah, if we are very sure our annotation pipeline worked really great, even we introduce the phage sequence in RVDB, and theoretically, if annotation pipeline works really well, those phage sequence we are going to include it will be annotated if there's any kinds of adapter or sequencing vectors. Yes.
Hi, I have a question for Alice online. It's nice to see a QC method already implemented. Can you share with us which negative and positive controls are you using? And also how you are storing which kind of raw data you are storing and for how long in the QC environment?
Sorry, Brad. So from a control perspective, Brad will be able to talk you through that and also the storage, because currently, Merck are performing the testing for us and storing all the data for us. So it's really around the Merck data retention policy.
All right, I'll start with the first one and not go near the data right now, but that's evolving. But in terms of controls that we use, so we'll run the sample through the method. Our controls right now are based on more of process controls. We validate in a modular format. We have expectations of the assay at each particular gate. So the way we work is that you do an extraction, then you have a gate to move on to the next step. After library preparation, you have a gate to move to the next step, and your sample has to pass certain criteria in order to do that. After sequencing, we also have a QC check at the end of that. So we check throughout the entire process. So they're more process-driven controls right now. For negative controls, we actually implement the use of reagent-only runs in the laboratory. And so we'll do those on a periodic basis. Generally, it's once every couple of months so that we understand what the background is from all of the kits and reagents that we use. So that way, if we do see any kind of positive signal, and of course we see signals, we need to do our secondary qualifications. One of the first things we look at is the reagent run. Is this related to the reagent run? Are we hitting the same sequence and that kind of stuff? So that's the controls right now that we have along with this particular assay. Now for data, that's a loaded question. I will say that to date, we have not deleted or removed any NGS data. We've been saving it. We've had assays online to GMP since 2016. We have 500 terabytes of NGS data in our repository. And I did learn that the next thing after terabyte is petabyte. I don't know that I actually wanted to know that, but I do. So we are looking, as a CRO, we're looking at other options in terms of data retention because we ask ourselves the question, is it the CRO's responsibility to maintain the data, or is it the sponsor's responsibility and the license holder to maintain the data? Now, that's all embedded in quality agreements, which of course we need to agree to with our clients in advance, but it's something that we are looking at right now as an organization, but we don't have a defined plan. Allison, I don't know if you want to say more on that. I'm trying to be careful.
You've done very well, Brad.
Thanks.
Yeah, it's a huge amount of data to store, and there's a limit. And we obviously know we will hold the material for the length of the product life cycle or as agreed, but that comes with challenges as our technology becomes more extensive and we need more data storage. So I think I would probably just concur with Vlad. It's a conversation that is ongoing around how best to manage that clearly with our customers in mind. Life cycle.
And you are storing the original raw data, not only the FASTQ? Or
yes.
So right now we keep everything, but we're considering the FASTQ based on some of the conversations yesterday. Like, would you ever go back to the image file, even if the technology was updated? You generated it with an older technology, right? So we're considering the FASTQ files generally as the raw data, but we are saving everything right now.
Policy is to hold everything at the moment. All those policies are updated. Sorry, that will be what happens currently.
thank you.
One question from my side, which I was discussed, so coming back to the standards and to the transcriptomic approach and to the reference materials. So of course, transcripts for me seem to be some kind of pragmatic approach. On the other hand, we have also the concept in mixing these infected cells and so what do you think is superior, or what is the reason behind mixing the cells? I have to say my concern is a little bit it's a very difficult thing having living cells for standardization, because the cells might express a certain level of RNAs, and depending in which lab you are starting them to culture, would it vary, or have you investigated that, or what is the experience in exchanging infected cells?
In fact, we are not mixing the cells, we are mixing the RNA extracted from the cells, and indeed we have controls that are frozen and always the same. So it's not mixing the cells at each run, for example, not at all.
But the cells, the RNA would be produced in each site differently. So each lab would get the cells and make their own extract and their own normalization between the transcripts and the cell numbers.
Each lab has to make its own validation indeed, but if cells are distributed by an external laboratories as a frozen RNA, for example, which is perfectly possible, it could be a standard as well.
So actually, that's a great question, and the infected virus-infected cells that Sandra has displayed in the poster are sort of towards that goal of characterizing these cells in terms of, the copy numbers, and we've got different expression levels, but to provide them, as a one-time, or, it'll be vialed cells, and for direct use from the vials without expansion or any other changes. So they'll be provided, in that manner, and basically they'll be characterized for the expression after the vialing, before and after the vialing. And we've been monitoring their stability of expression, and it seems pretty stable even in cell culture. But the purpose would be not to expand the cells, but to use them just like the model viruses directly from the cell, from the vials. Otherwise, yes, you change the properties.
Well, we have done this kind of thing for the transcriptomic collaborative study.
Yeah, little self-promoting also for the poster for the last question or the previous question about in-process controls. You can look at a poster in the poster session. And Alice, maybe this is not a question for you, but I direct it at you. Could you just roughly describe the 32 days that your process takes?
Do you just want to take the the mic, Brad? So the reason being is obviously, Merck are our CRO, and they do the testing for us. So Brad is best placed to explain his 32-day turnaround time.
So I'm happy to provide the breakdown maybe later, but in general for GMP assays, when you consider a GMP process and that we're not doing R&D, right? So there's a lot of paperwork, a lot of processing. So it takes 2-3 days to get NGS through the lab onto the sequencer. You've got your extraction, you've got your QC, that kind of stuff. Once you're on the sequencer, you can generally run 19 to 29 hours if you're using P2 chemistry or P1 chemistry on the Illumina system, right? But then after that, there's some data migration that needs to happen. Then it goes into the cloud, and we need to perform the data analysis. Then it comes back, and scientists need to perform that secondary qualification. Once that's done, we need to write a C of A, and it needs to go to the quality team for a full quality audit, because this is a GMP assay and that takes time. So this is all what's considered in the turnaround time. But I will say our standard assay turnaround time is actually 42 days, but we gave Alice a special deal because of our collaboration and forecasting. Well, it comes down to forecasting, right? I mean, a lot of that turnaround time considers that a sample lands on my doorstep and I don't know that it's coming, which means now I need to get it into the lab schedule as well. So that's all sort of accounted for within that. Sometimes it'll sit for a week if I don't know that it's coming, but Alice and her team are really good at providing the forecast to us. And so that's why we're able to offer that either same day initiation or next day initiation when those samples come in. And we actually are trying to optimize that turnaround time a little bit more and moving to, I'll announce it, Alice, 28-day turnaround time. So we're continuously looking at that process to seeing how we can move a little bit quicker while maintaining quality and offering a TAT that's sustainable for our clients, because that's important. In a vacuum, yeah, we can probably do it in 10 or 14 days, but we can't do it continuously like that.
Thank you.
Non-target analysis.
Right. It's non-target agnostic analysis.
Hi, it's David here. I'm from Hudou Center Biotech, China. I have some questions for Peju. So thank you for the detail about how you generate the RBDB. It's crystal clear. But I'm wondering, have you done some maybe benchmarking comparison for using this new list RBDB compare than maybe the older one or other, like the GenBank or RepSeq, like maybe using the sequence data from the W panel. So after using the same pipeline, but you replace the database with the latest RBDB, and does the those NGS signal have a better correlation with the spiking concentration or not? And I mean, maybe sometimes we have a sequence data. There must be a group of our virus that have a very weak NGS signal. And if use this latest RBDB, will those I would say background noise be less than using other database? Because I mean, if this database provides a better performance, I think you don't have to promote it. We will all be using it for the biometric pipeline.
Yes, we have this kind of the performance benchmark. Years ago, when RBDB was released to public, we do compare with the regarding of the speed, performance, and also the sensitivities. And I'm not aware of your comment saying that you say that you use RBDB and compare with maybe some public domain database. You think the public domain database will have higher sensitivities? Is that?
I mean, because we don't have like the standard data, but right now we have the data from the panel, right? We have the panel for the 7 virus. So if using this new RBDB, will the NGS signal maybe like for the risk count or other signal you are using has a better correlation result with the spiking concentration?
We didn't have this kind of the benchmark regarding of the correlation of the spiking number versus the hit you have, because that is not the purpose for RBDB. Yes, I need to emphasize one thing. Sometimes so that's why it's very important you have the follow-up if you use the public domain databases, because by any occasions you think you got more sensitivity or you have more mapping rate by using the public domain database without the curation versus RBDB, you got higher hit. You better do the follow-up, because RBDB by that scenario definitely will get you less hit, but at least this kind of reduction of hit probably because whatever the rate you map to the public domain database, they are most likely because of the non-viral sequences, such as host. So that's why I emphasize that, yes, RBDB, you heard lots of talk today. We are talking about target analysis and non-target analysis. Also, I described in yesterday's workshop. So for the non-target analysis, yes, when you see something in your primary screening, don't be shocked or happy. You need to confirm the hit you have by the counter screening.
Yeah, right. So so that's the second question. Like, so if we don't have a better correlation with the the spiking concentration, which is expected, but is the background noise also decrease using the RBDB?
Yes. Actually, we didn't have this kind of benchmark, but I remember 2 years ago in IABS in Rockville, one participant, they definitely compare the public domain database and RBDB, and they definitely see higher specificity, which means you will have much lower background by using RBDB. And I don't know, because we have lots of RBDB user here, maybe you can talk to them and they can share more experiences.
Okay, thank you.
There's no urgent question
Yeah,
anymore. So
I have a quick follow-up question for
quick
Brad.
follow-up and then.
Yeah. So the negative control sample, or it's like environment monitoring sample that you mentioned that use testing reagent only. Are you able to get enough nucleic acids from just testing reagents to a sequenceable library?
So we are, because we're using an intermediary, it's an agnostic amplification technique. We use Genomify to amplify. It's a rolling circular based amplification. So we extract essentially from water, and then we'll perform that. We'll perform the cDNA and then amplify from there. So you do need to beef up, because you don't get enough. I mean, if you really wanted to, we're also considering adding additional maybe controls into that so that you do have enough nucleic acid. But right now, that's where we are.
last comment. I know it's
So
getting late.
Yeah, so, and of course, because we've got two CROs here, the question is obvious. I mean, as as everybody has talked about, there's an increase and uptick in terms of use of NGS, and therefore you guys may be, more, I guess, in demand or, for performing, with a short turnaround time, but how will you manage the increased workload and still meet the expectations?
It's, so, so capacity expansion is something that we've undergone over the past several years. We actually just had another major capacity expansion in our Rockville facility. When we built out those labs, we took all of the contamination mitigation under advisement there. So we have rooms for library preparation. We had one previous room, but now actually we have one room with 4 sub-suites so that we can actually process samples simultaneously in the same space, because you can't, we've done this before, we've been down this road, you can't process 2 different backgrounds together, especially if they're high titer different viruses. You will see one background in the other, whether it's index hopping or procedure or anything like that. And so we've taken stuff into consideration like that in order to scale our processes. And our latest capacity expansion puts us at a pretty large capacity to handle samples. And I guess we'll have to monitor and evaluate as we move through. We're doing that at multiple sites. Mark, I don't know if you want to.
Regarding the wet lab, we have two sites, one in France, one in the US. And in the US, it's a large laboratory with plenty of room, so it's not really a problem. And regarding the bio-IT, we have made a lot of efforts to increase the speed of calculation without consuming too much resources. So our pipeline is quite fast now, much faster than previously, and it's no more an issue, in fact.
Okay, thank you. Then I think it has been a fruitful day, great discussion, long day, and so I wish everyone a good evening and happy to see you back tomorrow. Thank you.